Okay, so our final speaker, Professor Doug Wallace, is arguably been the most prominent and recognizable figure in the field of mitochondrial biology over the past, say, 35 years. So I also like to point out when I give mitochondrial talks that in the 50s and 60s and 70s, and really it was the heyday of mitochondrial bioenergetics, including a lot of work that was done right here at UW-Madison. After that, after people sort of came around to Peter Mitchell's chemiosmotic coupling hypothesis, and he won the Nobel Prize for that in '78, there was a sense in the field that mitochondria really figured out. And there was a bit of a lull of sorts after those major discoveries. And Doug is really someone who I think of who, like in this post-Oxfos era, really shepherded mitochondria through that, and through some really seminal discoveries, I think, helped set up the kind of renaissance of mitochondrial biology we're experiencing today. And a lot of that really came through discoveries such as his discoveries of maternal inheritance of mitochondrial DNA, for example. One of those facts we sort of all know about mitochondria. It's really one of Doug's discoveries. The discovery that mutations in mitochondrial DNA can give rise to many different metabolic disorders and can contribute to things like cancer and aging. And also the use of mitochondrial DNA variation to reconstruct the origin and ancient migrations of people, of women, given the maternal inheritance. Revealing that humans arose out of Africa some 200,000 years ago and left Africa some 65,000 years ago to colonize Eurasia and then eventually the Americas. This all came out of Doug's work. He's also written some of the most comprehensive and well-cited reviews in the field, including his 2005 annual reviews paper that really ties mitochondrial dysfunction to so many different diseases. That's been cited some 2,000 times now. So Doug originally got his BS at Cornell in genetics and developmental biology. Did his doctoral and postdoctoral work at Yale. He's had faculty positions first at Stanford and then Emory, UC Irvine, and is now the director for the Center of Mitochondrial and Epigenomic Medicine at University of Pennsylvania. He's also the Michael and Charles Barnett Chair of Pediatric Mitochondrial Medicine and Metabolic Disease, and a professor in the departments of Pathology and Laboratory of Medicine. So given what I just said, it's not surprising that Doug is a member of the American Association of the Advancement of Sciences, the National Institute of Medicine, formerly the Institute of Medicine, the National Academy of Sciences, and as recently a recipient of the prestigious Gruber Prize in Genetics. So please join me in welcoming our final speaker, Professor Doug Wallace. Thank you.
Well, I want to thank very much both Jerry and David for all their work putting this conference together and for inviting me and for having the privilege of being in the same program as Jody and Vamsi, who I respect enormously. I'm a clinical geneticist, and I've always been interested in patients. And one of the interesting things about patients that come to see me is that they have all kinds of problems, many, many complex problems. So that has led me to ask the question, why is it that we don't understand the common complex diseases and why can't we treat them? And what kinds of clinical problems do these patients present with? Well, they present with neuropsychiatric problems. They present with heart and muscle problems, visceral, metabolic, inflammatory problems, even cancer, and of course aging. But what's interesting about those particular kinds of phenotypes is that those are, in fact, the common phenotypes that are important to our general population's health and welfare. And there's a very disturbing situation right now in Western medicine, and that is that there's an inverse proportionality between the frequency of disease and our ability to understand it and treat it. So the common diseases we don't understand and we can't treat, and the very, very rare diseases we sometimes can understand and treat. But this, of course, does not make the American public very happy, and that's, of course, a big problem for us. So that really raises the question, why is it that we aren't doing more successful at addressing these problems? And I began to think about this a number of years ago and began to think that, well, maybe the problem is not that we aren't working hard enough or we don't maybe even have enough money, although we certainly could use more money, but maybe the problem is that we were looking at the problems in the wrong way. And what is this idea of looking at the problems in the wrong way? Well, when we go to school, we're told things, and we're told things when we're very young, and we then believe them, and they then form the basis on which we look at the world. And, you know, that's why many people stay the same political party for their whole life, or the same religion, or so on. And these same ideas are relevant in science. So we're taught things to believe, and those ideas, which are called paradigms, they form the intellectual basis on which when we interpret other things we see in the world. The problem with their paradigms is not that they aren't really powerful at predicting the future, but that they are human constructs. And in fact, we may not make a human construct that completely explains the real phenomenon in the world. That is, we don't know what we don't know, so our construct can only reflect what we do know. So then it occurred to me, well, maybe the constructs we were using were not adequate to address these common problems. And so that then led to the question, what are the biomedical paradigms that we base all of our research on, and are they adequate to the task? And there I really broke them down into two concepts. One, what I call the anatomical paradigm of disease. And this goes back to Vesalius, who worked about a half a millennia ago and was the first to really define the anatomy of the human body. And after that, that was so compelling that almost all physicians then decided to learn more about each organ system. And that led over the next 500 years to ophthalmologists, neurologists, cardiologists, nephrologists. That is, most people then specialized in an organ system. And all of the NIH system centers are all about organs. So that idea then has a corollary that's unspoken when we learn about medicine. The corollary is that if you have a symptom in an organ, then the assumption is that there is a problem with that organ. That is, if I get a headache, then I go to the neurologist, and he or she is going to tell me why I have a headache. The other major idea, of course, is less old, but no less compelling, and that is that genes are inherited according to the laws of Mendel. That's about 125 years old. And we believe that inherently. But we also know now that these genes are on chromosomes, and it's the chromosomes' behavior that creates the inheritance of the nuclear genes. That is, each of us has 2 copies of each nuclear gene. Each of our gametes gets 1 copy, and then the next generation, recombination of the sperm and the egg, get 2 copies again. And so that idea leads to a corollary also, and that corollary, which I was strongly taught when I was early student, is that if something is inherited according to the laws of Mendel, it's genetic, and if it's not, it's environmental. And there's even an equation that I had to memorize and take my clinical boards showing that I understood that idea. Okay, but with all the power of these particular ideas, they haven't been very successful in understanding these common diseases. So what could be missing? Well, in fact, interestingly enough, all the anatomical genes are encoded by chromosomes. So every anatomical event is, in fact, Mendelian. And how did that become so compelling? That was due to the Drosophila geneticists at the turn of the century. And they were looking at how anatomical aspects of Drosophila were inherited, and they were all Mendelian. And that then led to the extinction of the people that had a very different idea, that is, genes were more quantitatively distributed. But in fact, life is not just about anatomy. It's about being animated. And you're the most animated thing in my environment. And half a millennia ago, a guy named Newton realized that matter is inanimate unless it's acted on by energy. So if you're really animated, then mass is not the only thing that's important. Energy is also important. So we have to think then that life is an interaction between anatomy and the flow of energy. And of course, you need information for anatomy and energy. So this then led to 2 other corollaries, or ideas. One, that there could be a bioenergetic basis of disease. That is, you could have a partial defect in energetics, and it might cause symptoms in specific organs. And that there might be, in fact, genes that were non-Mendelian. And those genes, as it turns out, are the energy genes, the most important energy genes. So with these 4 ideas, then, maybe we'll be more successful at looking at these problems. And as you heard, this dichotomy evolved through the association of 2 different life forms. What is an archaeobacteria, we believe, and an oxidative bacteria that came together for various reasons, and they formed symbiosis. And the archaeobacteria ultimately gave rise to what we now call the nuclear cytosol, and the oxidative bacteria are the mitochondria, just as you've heard. Now, each cell then has hundreds to thousands of mitochondria. Now, what's interesting about that is a cell is limited by its energy. So a bacterial cell uses most of its energy to replicate DNA, transcribe it into RNA, and translate it into protein. So, in fact, the amount of genes that a bacteria can have is actually controlled by the amount of energy it can generate. And that energy generation is defined by its membrane surface area. So, in fact, the original nuclear cytosol bacterium and the mitochondrial bacterium had roughly the same genomes. Now, when they formed the symbiosis, there was, of course, a gene exchange between the 2 organisms, because that's what bacteria do. And in fact, there was an advantage to transferring genes from the oxidative bacterial genome to the archaeobacterial genome. Why would that be? Because if you transferred one gene from the mitochondrial DNA into the nuclear DNA, then 2 copies of that gene could serve all of the bacteria. So you then had a 1,000-fold savings in energy. So there was a tremendous genetic and evolutionary advantage to transfer genetic information from the oxidative bacteria to the nucleus, so that you, in fact, could free that extra energy to make more genes in the nucleus. And now we have 20 to 30,000 genes. So then the mitochondria and the nucleus became specialized, with the nucleus cytosol becoming specialized in anatomy and the mitochondria in energy. And how does energy flow? Well, energy flow, at least in the terrestrial cell planet land surface, through the flow of sunlight. And sunlight photons impinge on symbiotic bacteria in plants called cyanobacteria, which we also call chloroplasts. They pick up that photon of energy, use it to split water into hydrogen and oxygen. The hydrogen is fixed onto carbon to give glucose, and the oxygen is released into the atmosphere. And of course, that's why we have 20% of oxygen in the atmosphere. And so plants then are polymers of glucose. And then we have exterminated throughout the whole Midwest all different life forms except for wheat, corn, barley, and so on. And why do we do that? Because we're trying to harvest the sunlight. In fact, one can make a very good evolutionary argument that the dominant form of life right now on the planet are wheat, corn, barley, and so on, and rice, and that they have domesticated a bipedal primate to take care of their gene transmission. So anyway, we now have killed off all the wheats and have generated a lot of muffins. And so we then eat the muffins, and the muffins go into our bloodstream, and there the glucose is split into 2 molecules of pyruvate. The pyruvate enter a symbiotic bacteria, and the symbiotic bacteria then takes the hydrogens off the pyruvate, reacts it with the oxygen that you're currently breathing to give the water back and generate ATP as a chemical source of energy and heat. So in fact, the interesting aspect of this idea is that most of the energy flow is not about the nuclear cytosol, but about symbiotic bacteria, and yet only about 4 or 5% of the NIH budget studies that. Okay, so this is a biochemistry lesson from hell. I've taught biochemistry for almost 40 years. I've never found a medical student that liked it, but I love it, and so here we are. This is your outer membrane, intermembrane space, intermembrane, and matrix. Here's glucose going through glycolysis. Pyruvate can get reduced to lactate, or it can get an amino group to give you alanine. Pyruvate goes through pyruvate dehydrogenase to make acetyl-CoA. That drives the TCA cycle, the purpose of which is to strip the hydrogens off the hydrocarbons to put it on the carrier NAD. So now you have the reduced form NADH. That's oxidized by NADH dehydrogenase. The electrons flow through complex 1, or NADH dehydrogenase, CoQ, complex 3, cytochrome C oxidase, 2 electrons, reduce an atom of oxygen to give you a molecule of water. Now the problem is that you can't store electrical energy flow, and that's of course a big problem in the electrical net. So how did biology solve this? And according to Nick Lane, this was the first solution in biology, and that is it converts electron flow into a capacitor. And it pumps positive charges from the complex 1, 3, and 4 from the matrix into the intermembrane space, creating electrochemical gradient, and that's of course what Peter Mitchell won the Nobel Prize for. So that then electrochemical gradient can then be used as a source of potential energy for everything you want to do. You can convert it into ATP by complex 5, the ATP synthase that uses the energy to condense ADP and phosphate to ATP, goes out through a protein called the adenine nucleotide translocator, out through the voltage-dependent ion channel, and ATP is hydrolyzed to do work. ADP goes back in to be rephosphorylated. So if you do more work, you get more ADP. The ADP is rephosphorylated at the expense of the electrochemical gradient. You then mobilize more fatty acids and carbohydrates to generate more reducing equivalents to restore the electrochemical gradient. So you've coupled oxidation with phosphorylation, hence oxidative phosphorylation. Now everybody in this room has a different efficiency at pumping protons out and converting them to ATP. And we call that the coupling efficiency. So people that are very efficient at pumping protons out and converting them to ATP, they will require the minimum amount of calories for the maximum amount of ATP. And every time you burn a calorie, you generate heat. So they will generate the minimum amount of heat for the maximum amount of work. On the other hand, an inefficient coupling system will require more calories to pump the same amount of protons and convert them to ATP. So now you'll need to eat more calories for the same amount of ATP, but you've also burned more calories, so you generate more heat. So there's the coupling efficiency that allows your system to regulate thermal and work systems by this mechanism. The mitochondria also generate reactive oxygen species. This is an unpaired electron on O2. It wants another electron, that's superoxide anion. There's an enzyme manganese SOD that takes 2 of these to make hydrogen peroxide. If it sees a transition metal that's reduced like iron, it will get another electron, that's hydroxyl radical. That will also pull electrons out of lipids, proteins, and they damage the mitochondria. So excess oxidative stress will preferentially damage the mitochondria. As you heard from the elegant talk of Vamsi Mutha, the mitochondria regulates cytosolic calcium. And also it has this structure, the permeability transition pore, which is the self-destruct system that Jody mentioned. And this involves, we believe, the TSPO. It's regulated by the adenine nucleotide translocator. This component, according to Paolo Bernardi, is a part of the ATP synthase. And this can then normally be a closed door, but when the membrane potential is low, high energy phosphates are altered, ROS increases, or calcium load gets high. This then gets acted, it pops open, and it releases, it short circuits the membrane potential, releases the calcium, just as Dr. Mutha said, and that causes fluids to flow into the high osmotic strength matrix. The inner membrane swells, backs and back form a megachannel, release these stored proteins. They then create the apoptosome and the nucleases that degrade the cell from inside out. So why go to all this trouble? This is a high ATP demanding process. Well, because, as you'll see, this is a perfectly fine bacterium. And if that bacterium were just allowed to be released into the cytoplasm, as you will see, it would give an immune response. So the mitochondria that are central to energy, redox regulation, reactive oxygen species, calcium, apoptosis, and intermediate metabolism makes most of the high energy components, ATP, acetyl-CoA, alpha-ketoglutarate, S-adenosylmethionine, that are all involved in regulating signal transduction and the epigenome. I just want to mention there are several isoforms of the ANT. Okay, so basically we're 2 life forms, the nuclear cytosolic life form, which now has over 1,000 nuclear genes that function in the mitochondria, and the mitochondrion with its mitochondrial DNA that's transcribed into transfer RNAs, ribosomal RNAs, and the mitochondrial ribosome is translated using N-formyl methionine, just like a bacterium. It's sensitive to chloramphenicol and aminoglycosides, and it makes 7 of the 45 proteins of complex 1, 1 of the 11 proteins of complex 3, 3 of the 13 proteins of complex 4, and 2 of the 17 proteins of complex 5. So if you've put 1,000 or more proteins in the nucleus. Why keep 13? And I believe, and this is a Doug Wallaceism, I believe that the reason we have those 13 proteins in the mitochondrial DNA is because they're all the electron and proton wires of complex 1, 3, 4, and 5. And why would that be important? Because they all share the same substrate and product, the membrane potential. So if any one of these became leaky for protons, it would short the capacitor. And if the capacitor collapses, then you stop breathing. And when you stop breathing and you have no potential energy, you become inert, and we call that dead. Okay? So these proteins must co-evolve to maintain a balance of all of those 4 enzymes. And the only way that that can be done, if recombination were allowed, then my very loosely coupled mitochondria might be combined with Vamsi's very tightly coupled mitochondria, and we might have a mismatch of the different complexes and become very weird. So Mother Nature, being a woman, had a great idea. She decided to have the mitochondrial DNA with the wiring diagram of the power plant inherited from one and only one parent, and of course, being a woman, she decided it should be a mother. So it's transmitted from mother to all of her children. The mitochondria from the daughters to their children. Mitochondrial DNA of fathers go into the egg. They're seen as foreign and are selectively degraded. So the men have been thrown out for 2 billion years. It's appalling. But anyway, each cell then has thousands of mitochondria, and they're replicating inside your cells all the time and being gobbled up by mitophagy. So your mitochondrial colonies are in a steady state of growth and degradation. And as they replicate, they can accumulate mutations, and now we have a mixed population of mutant and normal. We call that heteroplasmy. The cell divided down the middle, then both daughter cells would have some mutant and some normal. But if the cell divided this way, then this cell would have only normal, and this would have twice as many mutant. So a heteroplasmic zygote can give rise to organs and tissues with different percentages of mitochondrial DNA mutant by a stochastic process. And when the percentage of mutant gets high enough, the energy output falls below the minimum for that organ, and then you have the equivalent of a metropolitan brownout, and that gives you a clinical phenotype. The mitochondrial DNA is then 16,569 base pairs, codes for a small and large ribosomal RNA, 22 transfer RNAs that punctuate the genes, ND1, 2, 3, 4, L4, 5, and 6 for complex 1, cytochrome B for complex 3, CO1, 2, and 3 for complex 4, ATPase 6 and 8 for complex 5. So there are 3 classes of potentially pathogenic mitochondrial DNA mutations. One is new maternally inherited mutations. So this mutation in the tRNA leucine gene at 3243, if you are 20-30% heteroplasmic for that mutation, then you will get type 1, type 2 diabetes or autism. If you're 50 to 90% mutant, you get systemic neuromuscular degenerative disease. And if you're 80 to 100% mutant, you die of an infant of perinatal lethal disease. Mutation in the tRNA glutamine gene accounts for a very significant percentage of late-onset Alzheimer's and Parkinson's disease. Mutation in the tRNA lysine gene will give you a kind of epilepsy, myoclonic epilepsy. There are literally hundreds of these protein synthesis mutations. There are also missense mutations, like this mutation in the ND4 gene at 11778. That's a missense mutation. If you inherit that from your mother, you're fine until you're about late in your mid-teens, and suddenly, without warning, you'll lose vision in one eye and then the other and be legally blind. That's called Leber's hereditary optic neuropathy. Mutation in the ND6 gene at 14484, same phenotype. Mutation in the ND6 gene at 14459, which is more severe. If you're heteroplasmic, you get optic atrophy. If you're pure mutant homoplasmic, you get generalized dystonia, like Parkinson's disease. Mutation in the ATPase6 gene, 8993, at 70% mutant gives you salt and pepper retinitis pigmentosa, 85% olivopontocerebellar atrophy, 100% death. Okay? So there's a quantitative effect for this population of bacteria inside your cells. There are also all kinds of cancer mutants, which I won't go into today. There are also ancient polymorphisms. Like this variant is found in 3/4 of sub-Saharan Africans. This variant is found in half of Europeans. That's lineage L. This is lineage H. A, B, C, and D arose in Central Asia, crossed the Bering Land Bridge, colonized the Americas. And then you're accumulating these somatic mutations with age. And so they then give you an aging clock. So this then leads to the idea that, in fact, what's really important about medicine is the energetic anatomy. There are high energy utilizing tissues, and that's the brain. 2% of the body weight, 20% of the oxygen. So a 10% reduction in systemically of mitochondrial energy has a huge effect on the brain without affecting the toe. Other systems, heart, muscle, renal, endocrine systems are all high energy demand tissues. There's an energy storage tissue. White adipose tissue stores fat to make ATP. Brown adipose tissue stores fat to generate heat. There's an energy homeostasis tissue. The liver regulates blood glucose. Why? Because that's your connection with sunlight. And there's an energy sensing tissue. Pancreatic beta cells sense high glucose, send out insulin, and says, convert energy metabolism toward glycolysis. Or when you're fasting, low glucose sends out glucagon, and that's from the alpha cells, and that says mobilize fatty acids, burn them to get over the low energy environmental sources. So this then leads to this concept that if we take anatomy out of the center of medicine and put energy in, then suddenly all the common complex diseases have the same pathophysiology. So this is oxidative phosphorylation. If it's defective, then mitochondrial replication becomes impaired. That will give you a progressive accumulation of mitochondrial DNA damage. This can be altered by mutations in the nuclear DNA or changes in the epigenome. Alterations in the mitochondrial DNA, such as ancient adaptive or recent deleterious mutations, alterations in the availability of calories or how they're used, or toxins for the mitochondria. As mitochondrial energetics declines, you decline over time mitochondrial function, and that's aging. A partial defect in mitochondrial energy will affect the brain, heart, muscle, and renal and endocrine system. Those are all the tissue-related diseases. But change in energy, the furnace, will also affect how the substrates are used, and that then accounts for diabetes, obesity, and hypertension. The mitochondria are the most common bacteria in your body, and we think as the cell declines in energy, without energy, the mitochondrial apoptosis fails, the antigens are released, and that's why we believe all these diseases have an inflammatory component. And finally, cancer is all about changing metabolism based on availability of substrates. So this is an example. This was a pedigree we studied in the early 80s. This woman had lactic acidosis and short stature. These were the actual people we could examine. They all had lactic acidosis, growth retardation, dementia, stroke-like episodes, hypertrophic cardiomyopathy, cardiac conduction defects. The oxidative fibers were degenerating. The glycolytic fibers were fine. They had these abnormal mitochondria. It's called mitochondrial myopathy or ragged red fiber disease. Now, this family had a mutation in the tRNA leucine gene at 3243, and it was had 70% or more heteroplasmy, hence giving maternal inheritance of the disease, and all of these young people died in their late teens or early 20s of either cardiac conduction defects or status epilepticus. However, this exact same mutation at 10 to 30% mutation will give you autism, or accounts for 0.5 to 1% of all type 1 and type 2 diabetes throughout Eurasia, or if at 100% will kill you with Leigh syndrome or perinatal lethality. So the question is, how could exactly the same mutation give such totally different phenotypes? So to test that, we use a method that my lab developed in the early 70s, which is to take a cell that was heteroplasmic from the proband, remove its nucleus, take the cytoplasmic fragment, and fuse it to a cell whose mitochondrial DNA has been removed by a method that was developed by King and Attardi. So this is so-called a row zero cell. But now you have this common nucleus, but now you can clone the cells and get a full range of different mitochondrial DNA genotypes or heteroplasmy levels. And so then you can look at these cells physiologically. And as the percentage of mutant mitochondrial DNAs increases, the ability of mitochondrial DNA-encoded polypeptides declines linearly. And this then affects the respiratory complex 1, 3, 4, and 5, those with mitochondrial-encoded subunits. There's an interesting aspect because, as Dr. Donardi indicated, mitochondria can fuse and fission, so mitochondrial mutants can complement each other in trans. So as the percentage of mutant mitochondrial DNAs increases, the respiration rate does not change until about 70%, at which point there's no longer complementation and respiration crashes. And that, I believe, is what happens with old age. These increases in percentage of mutant mitochondrial DNA change the mitochondrial morphology. So, in fact, the respiration rate is directly affecting the things that Dr. Nanarni mentioned. And one of the most remarkable things is that 30% mutant actually changes the volume of the cell by about 25%. And if we then plot the ratio of mitochondrial DNA to cell volume, you can see that at 20 to 30%, there's a maximum, then it goes to a minimum, and then it goes back up at 100%. Well, this is then diabetes and autism, this is neuromuscular disease, and that's death. Okay, so then we can do RNA-seq and look at how the genes are expressed. And about 20% to 30% of the RNA messages in a cell are mitochondrial DNA derived. But at 20 to 30%, that reduces by almost half. Then it slowly goes back up to about 90% where it recovers and then declines again at 100%. And if we look at the expression of the mitochondrial DNA encoded transcripts, 0% mutant, high, then a dramatic loss in 20 to 30%, then a rise to 60%, and then another loss at 100%. So there are four phases, 1, 2, 3, 4 of mitochondrial DNA transcription relative to the continual change of mitochondrial DNA percentage mutant. Now, if we look at the expression of nuclear-encoded genes, at 20 to 30%, glycolysis genes don't change that much. Only at 50 to 90% do they go up, and then at 100% they go back down. So what does that mean? That means at 20 to 30%, OXPHOS is low, glycolysis is not compensating. At 50 to 90%, OXPHOS is even lower, glycolysis is high, but at 100%, both are low. So we have four different states: normal, low OXPHOS, high glycolysis, low glycolysis, low OXPHOS. Okay? So that then directly relates to the changes in expression of the adenine nucleotide translocators. This is the ANT1 that's the most important in transporting out mitochondrial ATP. It goes up at 20 to 30% and then goes down. Another one just goes up and maximizes at 60. If we look at transcription of all the genes, 20 to 30% doesn't change. 50 to 90%, everything changes. And then at 100% goes back to low. If we start looking at some different genes, like these are the heat shock proteins for the mitochondrial unfolded protein response. You can see that they're induced at 20 to 30%, they go down at 60%, go back at 100%. Whereas the cytosolic heat shock proteins show the reverse effect. If we look at calmodulins, the calcium-regulated kinases, down at 20 to 30%, up to 60%, down at 100%. And the phosphatases are the reciprocal. If we look at the epigenome, DNA methylases, they're down at 20 to 30%, they're up at 60 to 90%, and they can go down at 100%. If we look at at the methyl binding proteins, MECP2 is the main gene that's mutated in autism, down at 20 to 30%, up at 60%, down at 100%. If we look at some of the unique histones, this histone is down, goes up to maximum at 20 to 30%, goes back down at 50 to 90%, goes back up to 100%. So what we're seeing then is a phase change in the expression of the nuclear genes relative to a continuous change in the mitochondrial DNA. So then we can take all of the transcripts and compute back to the transcription factors that must have been active. And at 20 to 30%, these are the transcription factors, positive and negative, that are affected. At 50 to 90%, it's a totally different pattern. At 100%, it's this pattern, and a cell with no mitochondria, it's yet a different pattern. Or you can do a principal component analysis and show this is normal. These are all the transcripts for diabetes and autism. These are the transcripts for neurodegenerative disease. This is lethal perinatal disease, and this is row 0 cells. So what the take-home lesson of this is a continuous change in mitochondrial energetics results in discrete nuclear epigenomic changes. And it's the nuclear epigenomic changes responding to the retrograde signaling that then creates the discrete phenotypes. That's why the same mitochondrial mutation can give totally different clinical phenotypes simply by changes in percentage heteroplasmy. Okay, so this is a family that has a mutation in the ND6 gene, proline to leucine. This was originally from Massimo Zeviani's laboratory. This woman was 50% heteroplasmy. She had optic atrophy, cerebellar ataxia. Her sister had only 5% mutant, but she had 3 different consorts. All of her children had 100% mutant and all died. So this is the kind of stochastic pedigree you can see with this maternally inherited heteroplasmic mutation. and this affects respiratory complex 1. So we made a mouse which has exactly the same mutation by making a cell line where we mutated the mitochondrial DNA and screened for the same ND6 proline 25 to leucine. We then take that cell, we enucleate it, take the cytoplasmic fragment, fuse it to a female embryonic stem cell that we've cured of its resident mitochondria with rhodamine 6G, replace it with the mutant mitochondria, put it in a blastocyst, put it in a foster female, breed the females for chimera and the chimeric females for transmission of the agglutine locus and the mitochondrial DNA. So when we do that, if we look at the optic nerve, this is the control, this is the mutant. We see swollen optic axons. We see demyelination, which is consistent with multiple sclerosis. And we see an age-related loss of the axons. If we allow these animals to age, we see the dopaminergic neurons of the basal ganglia selectively lost. We see a major effect on anxiety and depression. We see the motor disorders through both rotarod and pull tests and cylindrical tests, and at 2 years of age, we see significant learning defects. So a single point mutation in the mitochondrial DNA gives a generalized neurological disease. And we can look at the biochemistry by isolating the synaptosomes of the brain. Surprisingly enough, this defect only causes a 30% reduction in respiration rate. If we add ATP demands of the synaptosomes, we can drive that up to close to normal. That means the ATP levels are not affected. But what is dramatically affected, as we increase the demand of energy, we get a massive increase of reactive oxygen species. So we think this disease is due to oxidative damage to the neurons. So we can do the same trick again. Here we have made a mitochondrial DNA with a frame shift in the ND6 gene and a missense mutation in the CO1 gene. Put that into the mouse. The frame shift is due to the addition of a C in this rut of Cs that knocks the ND6 gene out of frame. But this particular embryonic stem cell, there was a reversion of 4% of the mitochondrial DNAs, deleting this T and restoring the reading frame. So now we had an embryonic stem cell that was heteroplasmic for the frame shift, but homoplasmic for the missense. We got one female mouse that was heteroplasmic for the frame shift and the reversion. And we could then take that female with about 47% frame shift, and we mated her, and all of her first and second generation offspring were 16% mutant, third generation was 6%, and fourth, fifth, and sixth, it was lost. If we take then the 16% females and mate them, they'll either give you 6% or 0%. So there's a directional loss of the frame shift mutation. If we superovulate the females, we find that we get the 16% or less, but never more. So this is a non-stochastic distribution of the frame shift. What does this mean? This means, since mitochondrial DNA distribution should be stochastic, it's a truncated distribution. What we believe is happening is inside the ovary, those cells, proto-oocytes, with the most severe mutants are selectively destroyed. So there is an intra-ovarian selection to eliminate the most severe mitochondrial DNA mutations. Why is that important? Because it has a very high mutation rate. If that didn't exist, the mutation load would be so great that the species would die. So we have then selection at the singular cellular level to get rid of deleterious disease. Okay, so then we've segregated the frame shift mutation to have homoplasmic CO1 mutation, 50% reduction in cytochrome oxidase. We get the ragged red fibers with the abnormal mitochondria. We get progressive cardiomyopathy with fibrosis. And amazingly enough, as the animals age, they get insulin resistance and type 2 diabetes. Okay, so the point of this is single point mutations in a mammal, either in complex 1 or complex 2, are completely sufficient in and of themselves without any nuclear change to give you all the clinical phenotypes of the common diseases. Therefore, these are bioenergetic diseases, and that's the proof. Okay? All right, so what happens if you combine two normal but different mitochondrial DNAs? So here we have a cell line with an NZB mitochondrial DNA. This is 129 mitochondrial DNA. We simply fuse the two together, and we now have a heteroplasmic mouse. So this is the founding female, that's the NZB, that's the 129. This is her daughter, NZB 129. The female transmits the heteroplasmy to her daughters, and their daughters to their daughters, as is seen here, whereas heteroplasmic males do not transmit maternal inheritance. Now this is 91 nucleotide differences. That's about the same difference as the extremes of you people. And they're scattered throughout the mitochondrial DNA, and they affect amino acids, tRNAs, ribosomal RNAs, and control region mutants, just like your variation. So we took these animals, back-crossed at 20 generations, and then we created a heteroplasmic animal that was pure for its nuclear genome. We then segregated some animals to homoplasmic 129, others to homoplasmic NZB, and maintains heteroplasmic animals. And then we compared the three. So if we look at, let's see what happens here. If we look at activity, this is the 129, night, day, night, NZB, night, day, night, and the heteroplasmic. They're about 30% reduced in activity. They also have about a 30% reduction at night in feeding activity. If we put the animals under a stressful environment, the NZB and 129 animals will soon give up. That is, they can only take a certain amount of stress. But the heteroplasmic animals are hyper-excitable. So they're like manic depressive people. Okay? So then we can ask them to learn this test. Here is an open field with holes all the way around it. We have different colored blotches so that the animals can orient themselves. And below this little hole is a little black box, and a mouse wants to hide. So if we put the animal in the open field, it searches around until it finds the black box and jumps in, and we use that as our indication of how long it took it to find the box. So we now have 129, NZB, and then the heteroplasmic animals. And this is trial 1, trial 2, trial 3, trial 4, and all of them learn, but the NZB 129 heteroplasmic learns more slowly. But then we wait 2 days and we put them back in the test field. The 129 animal immediately jumps in the hole. NZB animal jumps in the hole. The heteroplasmic animals have completely forgotten. They have no long-term memory. So simply mixing two perfectly normal mitochondrial DNAs, that is bypassing maternal inheritance, completely creates a severe neurological defect of neuropsychiatric disorders and learning disability. Okay, so then we can go back to the human case. Here is a pedigree of a mutation in the ND4 gene, a missense mutation, that causes sudden onset blindness. And this man is related and went blind through this female, and this female to these males, this female to these males, etc. So there's maternal transmission, but variable expression. And another interesting feature is there's a 4-to-1 male to female ratio, and that is very common in mild mitochondrial DNA diseases, and it's also exactly the same in autism. So the question is, why is there variable penetrance? So it turns out that the variable penetrance is due to the mitochondrial DNA background that the mutation occurred. So these are 3 different mutations that will give you Leber's hereditary optic neuropathy. But you can see that they have different severities of complex 1 defects. So how can they have the same phenotype? Because the severe one is found, this is a lineage in Europeans that's 10%, and it doesn't matter what lineage this mutation occurs on, you'll still get optic atrophy. But the intermediate one, a third of the individuals are this lineage J, and the very mild one, more than 3/4 are J, implying that the background increases the penetrance of the mild mutation. And here's a mutation in the ND1 gene that gives exactly that same effect, and that is a tyrosine codon 30 to histidine mutation. So what are these lineages? These lineages are, in fact, changes in the mitochondrial DNA as women migrated out of Africa into Eurasia and the Americas. L0 is the founding lineage found in the Khoisan people of the Kalahari. L1 and L2 are pygmy lineages. L3 are sub-Saharan Africans. M and N arose in Ethiopia. M stayed in the tropics all the way down to Australia. N went immediately into the temperate zone, giving rise to lineages I, W, X, H, J, T, U, UK, and V. Then later, M evolved through alteration to some Central European types, including C and D. Then C, D, and A became enriched in Chukotka, and 20,000 years ago crossed the Bering Land Bridge to colonize the Americas. So here's something amazing. Nuclear polymorphisms are panmictic, that is, they just change in frequency. But mitochondrial DNA polymorphisms are highly geographically constrained. Why would that be? Because here in Africa, if you want to reproduce and be a woman, you need to run away from lions. That means you have to be tightly coupled to have maximum ATP per calorie used. But up here, the lions froze to death, so that's not a problem. The problem is you're going to freeze to death. So how's that get solved? By accumulating mutations that decrease the coupling efficiency. Now, that's making less efficient mitochondria, so you'll eat more calories for the same amount of ATP, but generate more heat. So this then is one of the adaptive tools that the mitochondria has allowed our ancestors to colonize the world. So out of Africa then, the lineage that moved into the temperate zone had 2 missense mutations, whereas the lineage that stayed in the tropics had only synonymous random mutations. And these affect membrane potential and therefore coupling efficiency. Every mitochondrial DNA lineage is founded by a highly functionally conserved amino acid change. So this lineage T changes an ND2 gene. It's conserved all the way to C. elegans. J1 and J2 are founded by cytochrome B mutations. This one is conserved all the way to E. coli, yet they're polymorphic in the human population. Here's a study we've been doing over the last 20 years in Tibet, and we sequenced the mitochondrial DNA for Tibetans at high altitude. Why high altitude? High altitude is low oxygen, and mitochondria, of course, use oxygen. What we found in the M lineage, which is found in Asia, is in fact this variant 3394 had occurred 3 independent times. It did not arise on the N lineage. If we ask what's the frequency of this lineage versus the altitude of the different villages we studied, we see that as the altitude increases, the frequency of the variant increases. And this particular M lineage also increases with altitude. So this has an odds ratio of 24 and implies then that this missense mutation is selected for on the M lineage with high altitude. So what does that do? Well, if we look at the N lineage, the macrohaplogroup N lineage, which is found in the temperate zone, then you can see that the T to C reduces the complex 1 activity in these somatic cell cybrids by about 7 to 20%. But the shocking thing, these are 2 N lineages found in Asia, and you can see that the T form is as different from the T form F to B as either the T or the C is for B or F. That is, both the background mitochondrial DNA and the missense mutation are important for the overall biochemistry. Moreover, if you look at the M lineage with the C mutation, which is deleterious on N, actually is very good on the N lineage. Here's another variant that's found. This variant is in tRNA glutamine, A to G. It's about 10,000-year-old European mutation. It's found in less than 0.4% of Europeans, but if you look at aged populations, it's 3% of Alzheimer's, 5% of PD, and 7% of ADPD. So here is a variant that is not deleterious during reproductive age, but if you live long enough, gives you neurodegenerative disease. And this is as much variance as all the nuclear gene variants combined, and yet this is never quoted. It's been repeated 10 times. So we also get unique variants arising. This 3397 ND1 gene, changing methionine codon 31 to valine, gives you franked ADPD. So now we have a very weird situation. We have this 3394C, tyrosine codon 30 to histidine, that increases the penetrance of labors on low altitude on the N lineage, but is adaptive to high altitude on the M lineage. And we have the adjacent codon, 3397, methionine 31 to valine, which itself is deleterious on the N lineage. So how could this be? Because here on the N lineage, we have these 2 missense mutations out of Africa and the tRNA glutamine gene. That gives you this lineage for Alzheimer's. But these 2 variants, when combined with these 2, this one plus this one is very severe. This one plus this one is milder, giving you labors, obesity, and diabetes. But on this lineage, where these do not occur, then this particular variant changes complex 1 subtly enough to be adaptive to high altitude. And what we think happens is a subtle change in complex 1 induces cytochrome oxidase, and that gives you low oxygen tension resistance. So we've now, we and many others, have correlated mitochondrial lineages with Alzheimer's, Parkinson's, macular degeneration, migraine, psychiatric disorder, stroke, diabetes, cardiovascular disease, sepsis, AIDS progression, osteoarthritis, aging, cancer, athletic performance, and many, many other phenotypes. Okay, so what about nuclear mutations? This is a mutation in the ATPase6 gene. It's found in heart, muscle, and brain. This is a human pedigree. The mutation occurred about 500 years ago in Switzerland. This is a religious isolate. We have a lot of consanguineous marriages. These are double lines here of heterozygous nuclear genes. This is on chromosome 4. It's a nuclear gene. And these then give rise to individuals that are homozygous mutant. And this shows that it's a frameshift mutation with a deletion in one of the bases. So all these people get cardiomyopathy and myopathy. It's not surprising. What was surprising is some of them could live well into 30 or 40 years and have just hypertrophic cardiomyopathy, but others progressed very rapidly to dilated cardiomyopathy and required heart transplant. So why was there a difference? So what we did, we looked at then first the echocardiographic studies. The people that are wild type for ANT have very consistent heartbeats, and these are velocity vector imaging vectors, and green is normal. But the ANT1 mutants, you can see highly dysrhythmic and very severe alterations in heart contraction. The question then is, why was there a difference in phenotype? So this is from a 15-year-old boy that we had heart transplanted. He's homozygous ANT1. And you can see that he has the pericrystal arrays, highly abnormal cardiac mitochondria. So what turned out to be the difference? All the people that progressed rapidly to dilated cardiomyopathy were European haplogroup U, and all the people that had prolonged normal cardiomyopathy, or just dilated hypertrophic cardiomyopathy, were haplogroup H. That is, the mitochondrial background completely modified the outcome of the homozygous nuclear mutation. Okay. So then we made a mouse. This is our mousicizer. We're going to exercise the mouse. This is the wild type. This is oxygen. This is CO2. The mouse starts exercising, starts blowing off CO2. Then it goes to the glycolytic type 2 fibers, starts blowing off CO2 to buffer the lactate. That's the anaerobic threshold. So a wild type mouse can exercise till the experimenter collapses. But a mutant mouse starts exercising and immediately just falls down. And that's because it has a proliferation of highly abnormal mitochondria, massive mitochondrial proliferation, hypertrophic cardiomyopathy, and it cannot get ATP out of the mitochondria to sustain the heart and the muscle. So if we now look at the echocardiographic analysis of these animals, wild-type animals, uniform heartbeats, both by the velocity vector and by just the heart electrocardiogram. ANT2 shows the same dysrhythmia. Okay? And if we look at ejection fraction, wild-type animals throughout life, pretty constant. The mutant animals decline, but note this tremendous variability in ejection fraction of the ANT mutants. So when we look at the ANT mutant mitochondrial DNA, so here what we're going to do is use 2 PCR primers, and we're going to amplify all the way around the mitochondria and then measure the length of the fragment. So with the wild type and heterozygous animals, we have full-length mitochondrial DNA and only occasional deletion. But the homoplasmic mutant, we have severe depletion of the mitochondrial DNA and massive increase in damage. And so that heteroplasmy is the secondary damage to the mitochondrial DNA that is eroding the ejection fraction. And if we look at heart transplants from patients with dilated cardiomyopathy, we also see between a 10 and a 2,000-fold increase in somatic mutations. So, to test then whether the mitochondrial DNA interacts with the nuclear DNA, we combined the ANT mutant with wild-type mitochondrial DNA, CO1 mutant, or ND6. So when you put ANT together with CO1, you don't get that much difference. These are the velocity vectors, so here and here. But if you combine ANT1 with ND6, you get a huge effect. And therefore, it matters what's the mitochondrial partial defect that interacts with the nuclear gene variant for the phenotype. So this just shows the echocardiograms, wild type CO1, ND6, ANT1, ANT-CO1, and if you look at ANT-ND6, it is completely dilated. And if you look at lifespan, the ANT-ND6 has a 50% reduction in lifespan, and all of the different mutants have reduced lifespan relative to the wild type. But there is clear synergy between ANT and ND6, and you get this very interesting progression of abnormal mitochondria. So why is the abnormal mitochondria relevant to cardiac effects? So this is a discovery that Megan McManus together with Martin Picard found. If you look at cardiac mitochondria, the cardiac mitochondria are in long lines. And what they observed is that at the interface of the mitochondria, there is this darkening of material, which they have called the intermitochondrial junctions. And if you look at the density on the outer membrane, then at the junction and the outer membrane, you can see the protein concentration is much higher. But the astonishing thing that Megan observed is that wherever these junctions occur, all the cristae align across the mitochondria. And so here we're looking at the cristae alignment where there's a junction and where there's not a junction. Or here's a lipid droplet and mitochondria around it living on that fat. And you can see that the cristae literally circle the lipid droplet. So what does that mean? That means the mitochondria are coupled. And the coupling of the mitochondria then allows all the membrane potential to oscillate in, we believe, this is our hypothesis, that they oscillate in sync. And that means that the power of the oscillation is multiplicative of the mitochondria in line, as opposed to mitochondria that were independent and then oscillating independent of each other. And so as the amount of energy demand increases on a tissue, and this would then be cancer cells, iPS-derived cardiomyocytes, soleus, diaphragm, and heart, the number of these interactions increases. And that, we believe, is a major effect on power output. Now, if you look at patients or cell lines with defective mitochondria, what happens? You can either get disorientation of the cristae alignment, or you can get the proliferation of abnormal mitochondria that breaks down the junctions. And we think both of these then reduce the power of the high energy tissues and are a major factor in the pathophysiology of the disease. So I'd like to finish then by saying that up till now, we've considered all the common complex diseases as related to nuclear genes. And literally hundreds of millions of dollars, if not billions of dollars, have been spent on looking for the nuclear gene mutations for all the common diseases. And of course, a significant 10 to 20% of the variance has been explained. I'm not saying that isn't real, but there's still a huge amount of variance that's not explained. Well, what does the nuclear cytosolic system do? It has a quantized genetics, it encodes anatomy and the anatomical genes of energy. But there's another genome, as both Vamsi and Jody mentioned, and that's the mitochondrial DNA. It's entirely about energy, and it has a quantitative genetics. The mitochondrial genome mutation rate is 3 orders of magnitude higher than the nuclear genome. Why can that be? Because here you can monitor a mitochondrial defect in a single cell. So inside the ovary of every female in this audience, there is a selective process that's finding those proteocytes with damaged mitochondria and killing them by apoptosis, so that only the best oocytes are ovulated and fertilized. So we can have a high mutation rate without high genetic load. On the other hand, a nuclear gene mutation has to go all the way through development before it can be acted on by the environment. So that's a tremendous waste of energy. So in fact, the nuclear genome mutation rate has to be low, because if it were high, the system would be deleterious. So the high mutation rate of the mitochondria with the intra-ovarian selection allows high genetic variability of physiological properties. And that variability then allows our species to rapidly adapt to environmental changes. And that then, I believe, is a major evolutionary driving force for radiation of species, and I believe also speciation. And I feel that it's the energetics that's important in the common diseases much more than the anatomy. Finally, these are coupled by the epigenome, but it's the mitochondria that are generating all the substrates to drive the epigenome. So why would that be? because the nucleus can't replicate its DNA, transcribe it into RNA, or translate it into protein unless there's enough energy. So it must know what the energy flux is through the mitochondria. And that then is done through these high-energy intermediates that regulate the epigenome and control the nucleus. So I'd just like to finish that this overview has been the product of work from a lot of people. Some key people that have been involved in the mouse work are Wei Wei Fan, Grant McGregor, Ryan Lin, Ryan Morrow, Mark Sharpley, and Katrina Waymeyer. Physiological studies, especially the anatomy with Megan McManus and Martin Picard, and then many other supporting colleagues. All of the Tibetan studies were done with the 3rd Military Medical University, Fuyunji, my long-term collaborator, and initially with Lorna Moore, who was at Case Western. The studies of the eyes of the ND6 mouse was with Alfredo Sedun, Valerio Corelli, and their colleagues. The studies on the epigenome with Paolo Sassoni Corsi. The family that we studied was studied from the Clinic of Special Children, sorry for that typo, Kevin Strauss being my main collaborator. Then all the population genetics went over many, many years with my colleagues in Africa, Siberia, and other places. Thank you very much. Yes, sir.
Can you expand a little bit on your points about the relationship between energy and as a driver of epigenetics in a more specific way?
Yes, exactly. The mitochondria generates much of the ATP, much of the acetyl-CoA. It generates formate and ATP, which is necessary to make S-adenosylmethionine. It makes alpha-ketoglutarate. It also generates succinate, fumarate, and a number of other reactive molecules. These are all the substrates for all the major nuclear epigenomic enzymes. So the idea is that mitochondrial energetics is changing the substrates that then drive the modifications of the proteins of the epigenome, which in turn regulate gene regulation. So this then is a physiological coupling between energetics and the nucleus. Also, I'd like to mention that calcium is another major important modulator of this. And people at Penn Animal Biology program have shown that subtle changes in the mitochondrial membrane potential increase the cytosolic calcium level, as Dr. Amutha mentioned, and that activates NF-kappa B, acts through a signal transduction system that activates specific histones that alter also the gene expression in cancer. So there's a very elaborate crosstalk between these systems, which we're just beginning to understand. Yes? Wow, that's a shock. Okay, but I was amazed that they wouldn't dispute me because I've been disputed for 40 years. Okay?
Maybe you're onto something. So, and the mouse studies elegantly show that this is possible. But when we move to the human being, we have a problem. And that is we don't know which way the causal arrow is pointing. Does the disease cause mitochondrial dysfunction? I'm speaking about neurodegenerative diseases where the correlation is very strong, but we don't know which way the arrow goes. Or do the mitochondrial problems play a role in the molecular etiology of the disease itself? So you brought up the GWAS studies. I wonder, do you have any evidence that mitochondrial mutations are substantial contributors to autism, Alzheimer's, multiple sclerosis, any of these, that we can account for more risk using mitochondrial DNA than we can using the GWAS nuclear DNA? Or is there still a missing component of risk?
Great. Well, let's start from the end and go back to the beginning. Okay. So yes, we do many association studies between mitochondrial DNA variation and complex disease. So we are just finishing up a paper on autism where we have done 2 different very large population studies, found specific mitochondrial DNA lineages that are highly statistically correlated with autism. The important thing, though, that is often missed in this area is that there's tremendous population stratification of mitochondrial DNA. And so when people do big GWAS studies, like they want to have 30,000 cases, they'll mix populations across Eurasia. Well, that immediately washes out all the significance. So what's interesting about the mitochondria is we can get statistical significance with, say, 300 to 500 cases, as long as we work within a well-defined stratified population. So I think that there's a number of conceptual differences about how nuclear variation is studied and mitochondrial variation is studied. And because of the way those studies are done differently, mitochondrial variation has been overlooked. The next thing we found is that in working on Alzheimer's disease, if you find mitochondrial lineages and then you stratify the cases by the mitochondrial lineage, you'll now find nuclear variants that were never found in the original GWAS. So getting back to that, I don't mean to imply that only mitochondrial DNA mutations are involved in these diseases. Parkinson's disease is a classic example where there are clearly autosomal nuclear mutations that cause Parkinson's disease. But every one of those that has been well studied, PARC, PINK1, etc., they all, in fact, destabilize the mitochondrial DNA. So remember, the mitochondrial DNA's survival and function is directly related to the nuclear genome. DNA polymerase, RNA polymerase, all the helicases, those are all nuclear encoded. So mutations in any of those genes will affect mitochondrial function. And I didn't even go into it, but we have many pedigrees. Well, I did mention the cardiomyopathy, but we have many pedigrees where there's a nuclear mutation and a mitochondrial lineage that interact to give you the severe phenotype. So I'm not trying to imply that nuclear genetics is not important. I'm a nuclear geneticist that got misplaced. I think that both are very, very important. What I'm trying to argue is until we also look at this other genome, we can't parse out all of this interaction. And so I think we're missing that middle ground. So yes, there's mitochondrial specific variation. Yes, there's nuclear variation, but it's the nuclear cytoplasmic interaction that's that big void that you were talking about. And so that's why our center is called Mitochondrial and Epigenomic Medicine. We're trying to overcome that void by this crosstalk between the nuclear and the cytoplasmic system. I hope that was close.
Well, I hope you left room in there for other factors in the environment.
Absolutely. The point is that, as I showed you with that circle, the whole left sector was availability of calories, how those are used, whether you exercise or not, environmental toxins, those are extremely important. One interesting hypothesis, why is it that autism, diabetes, and obesity are going up exponentially? Well, one thing you could do is blame McDonald's hamburgers. Okay, well, that's one thing. But there may be another idea, and that is we, in our effort to make monoculture of wheat, corn, barley, and so on, we are adding a lot of fertilizers and a lot of herbicides and pesticides to our food supply. Almost all of those are potent mitochondrial inhibitors. And in fact, FDA never looked at that because they only look at anatomical changes in the mice. But if the mice have altered sleep patterns or altered circadian rhythm, that was never in their protocol. But those are the things that are getting hit. And those are the things that are creating the psychiatric disorders, diabetes, and affecting energy flow, and therefore we think diabetes and obesity. So we believe that, in fact, it's an environmental challenge that's adding a stressor to the existing stratification of mitochondrial haplogroups that's accounting for this effect. And we're trying to actually look at Midwestern populations and ask, are there specific mitochondrial haplogroups that are more sensitive to these environmental stressors? And that might also be an aspect, but that's all ongoing.
Thank you.
Yeah. Yes, ma'am. Or did you want to?
Carolyn, how about your last
Okay.
question? And we'll,
You could make a hypothesis that it isn't actually the mitochondrial dysfunction that's causing all these problems, but the reaction to the mitochondrial dysfunction. What kind of compensatory
Yeah, that's a great question, and that goes back to the first point I made on the heteroplasmy with the 3243 mutation. What really surprised us is that for that mutation, with a continuum of percentages, at 20 to 30% mutant, we already can see that the mitochondrial DNA transcription rate is cut in half, and yet the nucleus does nothing. That's really astonishing. What it says is there must be thresholds by which the nucleus then decides, well, I can't wait anymore, I'm going to have to change. But once it decides to change, everything changes. So I think that there are these thresholds of transition. What we're very interested in is, could we develop medications that would allow you to get over these gap periods and therefore could eliminate diabetes and autism by stabilizing the mitochondria in this sort of, we call it the valley of death for mitochondrial function. So one of the things that people are very interested in is using nuclear transcription factor small molecule modifiers to alter mitochondrial function. And that's actually what's happening in diabetes therapy. So metformin inhibits complex 1, but what that does is it activates AMP kinase, and that turns on PGC1 alpha, and that upregulates mitochondrial biogenesis. Or you can have the rosiglitazones. They interact with PPAR gamma, a nuclear receptor, and that upregulates mitochondrial function. So I think just by chance, we're already finding ways to mitigate some of these systems by increasing mitochondrial biogenesis and therefore function. And I think that a lot of people are now getting very interested in small molecule screens to do that. And Jody actually has some nice work. And actually, Vamsi had a beautiful paper in Nature Biotechnology years ago showing that you could find some really exciting molecules by just screening for changes in mitochondrial function in cultured cells. Right? Okay, thank you all.